Hello everyone, welcome to our channel. I am Sridita Ghosh. I am your tutor for today's lecture. Today's subject is Food Microbiology, Part 1. I hope it will be very helpful for the aspirants who are preparing for GATE 2023 exam. So let's start. What is food microbiology? Food microbiology comprehends the study of microorganisms that colonize, modify, process, or contaminate and spoil the food. It is one of the most diverse research areas within microbiology and food microbiology too. So let's see a brief history about microbiology. Early food preservation. In 1900 AD, food poisoning was recognized. In 1795, Appert developed canning. In 1854 to 1864, food microbiology becomes a science. In 1900 AD, food poisoning recognized. Emperor Leo VI of Byzantium issued an edict that forbade eating blood sausage prepared by stuffing blood into a pig's stomach and preserving it by smoking. This actually caused botulism. Greeks and Romans in 1582, ergotism and claviceps purpura that was caused by rye. Food poisoning was recurrent because we didn't know what actually caused them. Appert in 1795, he collected food in jars and boiled to preserve it, but did not know why. In 1854 and 1864, Louis Pasteur finally discovered scientific basis for preservation methods. Food microbiology becomes a science. Pasteurization was actually first done in wine, cow milk, juices, etc. Why to study food microbiology? It provides clean, safe and healthy food to consumer, control of microbial growth, prevent food spoilage, prevent foodborne illnesses. Food preservation and production can be done in a very healthy and safe food method. Microbial characteristics. Food permits the growth of bacteria because it is high in nutrients, water, etc. So we need to control this growth in some circumstances. Microorganisms are commonly found in foods and the sources are first of all environmental, like from animals, dirt, feces, hide, digestive tract, etc. Fruits and vegetables, fertilizers, birds, wild animals, etc. Grain contains molds. What organism causes the most cases of foodborne illnesses annually? Campylobacter. What organism causes the most deaths due to foodborne illness? Salmonella. Groups of microorganisms. Bacteria, eastern molds, fungi, viruses, rickettsia, algae, protozoa. Morphologies, we need to have this in mind, like it can be rod-shaped, straight, curved, spiral, club, occi, chains, and clusters. The next topic is microbial growth phases. It is a very important question every year which comes in GATE, and questions are asked from every angle from this growth phase. Try to understand the growth phase properly. We can see here a graph which is plotted as CFU per gram. In the y-axis and x-axis we have time. So it means as the time increases, there will be growth in the microbial count. So we can see that there is at certain point of time, there is around lag phase is going on, that is marked as A, that is known as lag phase. This portion is lag phase. In the second portion, we can see the log phase or exponential phase. In this particular phase, the microorganism growth is very steady At a point of time, they will reach the highest level of growth. The second phase or the third phase is stationary phase, that is marked as C. In this phase, all the microorganisms stops their growth. They come at a stationary point. They will not show any growth at that particular point of time. And at last, when the time is consumed, at last this is D point in which there is death phase seen. Effects of pH, enzymes, nutrients. Other environmental factors are temperature, salt, age. No known pathogen grows below pH of 4.6. pH mainly affects two aspects, functioning of enzymes and transport of nutrients. No pathogen grows below pH of 4.6. C. Botulinum. pH less than 3.5 like citrus fruits, pickles, kiwi, condiments. pH 6 to 7 meat, milk, corn, vegetables, eggs. Molds and yeast more tolerant of pH extremes. Molds 0 to 11 and yeast is 1.5 to 8.5. Moisture. It is also an important factor like pH. Remove or bind moisture. Humectants, dehydration, water activity. Most fresh foods of water activity greater than 0.99 should have the of water activity 0.99 should it have the fresh foods only. One of the oldest methods of preservation is drying or desiccation. Humectants like salt, sugar, propyl glycol. Dehydration, it removes water and makes it dry. Water activity is the amount of water available for chemical reaction and all the microbial growth that takes place in a food. Pure water has water activity of 1, 22% of NaCl solution 0.86 and saturated NaCl solution of 0.75. Spoilage bacteria can grow in water activity of 0.91. Spoilage yeast can grow at a water activity of 0.88 and spoilage molds can grow at a water activity of 0.80. Microbial growth and water activity. Halophilic organisms can grow at a water activity of 0.75, xerophilic molds 0.61, as osmophilic yeast at 0.61. Lowest water activity of pathogen growth is 0.86. And one of the example is Staphylococcus aureus. Oxidation reduction potential, OR potential, it is also regarded as EH. It is the ease with which the substrate loses or gains the electrons. We all know that loss of electron is known as oxidized and gain of electron is reduced. Aerobic and anaerobic, microaerobic, facultative anaerobes. Others are nutrients, fastidious versus non-fastidious, biological structure, antimicrobial factors, naturally occurring factors. Extrinsic factors are temperature, relative humidity, gases in the environment, presence of other microorganisms. Temperature, microorganisms grow over a wide range of temperatures like psychotrophs, mesophiles, thermophiles, cytroduric, thermoduric. Other factors are relative humidity of the environment that can change the water activity. Environmental gases are carbon dioxide and ozone. Presence of other microorganisms. Competitive exclusion. It has general microbial antagonism and lactic antagonism. Thank you so much for watching our video. If you liked the video, please hit the bell button and subscribe our channel, and very importantly, share with your friends. Thank you so much once again.